Hey everyone! In this video, we're going to be talking about a formula called the Hardy-Weinburg equilibrium. So I'm going to go through some information about the formula and kind of the stipulations that are required for this to work, and then also kind of actually how you solve one of these questions. So the Hardy-Weinburg equilibrium is basically when, from generation to generation, the allele and the gene frequencies stay the same. So the numbers that we're going to be finding are the exact same every single generation, which means that the population isn't changing. So in order for us to have the Hardy-Weinburg equilibrium, we have 5 things that you need to know. Okay, you want to put a big star beside this because this is going to be a really, really, really important concept. So in order for us to be able to study a population, and it has to meet the Hardy-Weinburg equilibrium, it has to have these 5 things. So first, it has to be a large enough population that this works. So I'm talking like maybe 1,000 or more. Okay, we also have to have something called random mating, which means there's literally no reason as to why an organism would be mating with another organism. So when we're talking about having a mate that has something good, like maybe they're stronger or faster, or maybe they have a really nice coloration, or some sort of reason why the female would want to mate with the male, that isn't happening here. They're literally just mating randomly. So like plants, when we release pollen into the air and it randomly falls onto another plant, that would be random mating. Okay, it doesn't happen very often in nature because usually there's some sort of reason why the organisms are deciding to mate with each other. Okay, there also has to be a closed population, so no organisms can come in and no organisms can leave. And there can't be any sort of natural disasters that kill off a half of their population, which is what genetic drift is. So randomly a tornado comes through their population and kills half their organisms. That's not going to allow for this Hardy-Weinburg equilibrium to happen because then there's going to be changes in their population. And there's also going to be no selective advantage of a genotype. So maybe a certain coloration being better at camouflaging, that would be a selective advantage because they're better at hiding, they would get eaten less. Okay, which again doesn't happen very often in nature. It means that every single organism has to have the same chance of survival, which means that there's basically unlimited resources in the area, which again doesn't happen very often. So these 5 things are super important to remember, but again, it's really, really hard to find a population in nature that's doing all 5 of these things. So it's really, really not common for Hardy-Weinburg equilibrium to actually happen. But I'll show you guys how we solve Hardy-Weinburg and how we actually find those gene and allele frequencies. So we're going to start with some of the things that we're going to be using for alleles. Alleles, again, is like the dominant versus recessive allele. So in our formula, we're going to use P for the dominant allele, which would be like saying big A before, like big A is being tall. And then for our recessive allele, which would be like a little letter, we're going to use Q to find that. And then when we're looking at our genotype formula, because we have P being equal to our dominant allele, homozygous dominant individuals would be P squared because they have 2 of them. So big A individuals are going to be P squared. 2PQ we're going to use to solve for heterozygous individuals, so big A, little a. And then Q squared is going to be homozygous recessive. So our formulas are P plus Q equals 1 if we're just solving for allele frequencies. So how many of the individuals have the dominant allele, okay, versus recessive allele, we can use this formula. Remember, P is equal to big A and Q is equal to little a. And then for our genotype frequencies, P squared is the homozygous dominant, 2PQ is the heterozygous, and Q squared is the homozygous recessive. So when we're doing this, the reason we're doing this is because if there are no changes, that means that we're at genetic equilibrium, which means that we're stable. If we're at genetic equilibrium, that means that we're meeting the Hardy-Weinburg equilibrium. Okay? If there are changes over time, that means that the population may be evolving. Okay? And if a population is changing over time, we call that microevolution because it's just looking at a specific small population. So we call it microevolution. These points aren't in your notes at all, but you should write them down because it's important for later. So maybe just in the margin, that would be good. So we're going to solve some of these questions together. I'm just going to go through 2 examples with you guys, and then I'll let you guys do the rest on your own. We'll go through the answers later. So for example, the first question that you guys have, it says, in mice, black is dominant to white. So I'm going to start by writing down my legend. So if black is dominant, we would use a big letter to represent it, and white would be little b. We have a population of 400 mice, and only 36 of them are white. So if I know that they're white, I know that their genotype would be little b, which is telling me Q squared. So I'm going to figure out what Q squared is by dividing 36 by 400. And then once I've done that, okay, I got 0.09, which is Q squared. But in order for me to do anything, I have to find Q by itself. So I can use that formula P plus Q equals 1. So to find Q by itself, I have to square root both sides of my formula. This square root will get rid of the squaring, so I'm left with Q. And the square root of 0.09 is 0.3. So now I can use that formula P plus Q equals 1 to figure out the other allele frequency. So Q is my recessive allele frequency. Now I have to find P. And then once I've found both Q and P, I can put it into my second formula. So I'm trying to find P right now. So I have to have P by itself on this side of the equation. So I have to subtract Q on both sides so that when I solve for P, it would be 1 minus Q. So I take 1 minus 0.3 and end up with 0.7. So those are my allele frequencies that I just found. Now that I have both of those, I can use this second formula, Q squared, which represents my homozygous dominant, 2PQ, which represents my heterozygous, and Q squared, which is my homozygous recessive. We already know what Q squared is. We know that it's 0.09, but just to make sure that you're doing it right, you may as well do it again. So I'm going to put my information into my formula. So 0.7, which is P, and then I'm going to square it. And then 2 times my P and my Q, which is 0.7 and 0.3. And then also 0.3 squared. So I'm going to make sure that these individual numbers equal 1. So when I do all that math, 0.7 squared is 0.49. This is saying that my homozygous dominant individuals, there's 49% of the population that's the homozygous dominant. My heterozygous individuals, 42% of the population, and then 9% of my population is homozygous recessive. But I just want to make sure that if I add these three numbers together, that they do equal 1. If they don't equal 1, you've done something wrong. Okay, so you have to go back and fix your mistake. So just make sure that all of that equals 1. That's the whole point of this 1 being on the one side of the equation, just to make sure that it does actually equal that. So that would be the answer to our question, because it asks for the allele frequencies, which we've given right here, and then the genotype frequencies, which we've given right here. So let's try a second question together. So this time we're talking about the widow's peak. So it tells us that widow's peak is dominant and no widow's peak is recessive. And it tells us that 650 out of our population show the dominant trait. But when it tells us that it's the dominant trait, we don't know if they're homozygous dominant or heterozygous. So we can't use that information to start off. Instead, we always have to start with the recessive individual. So to figure that out, I'm going to subtract from my total population of 1,100. I'm going to subtract 650, and I get 450 left over, which would be my recessive individuals, which would be little n, little n. Okay, that's telling me Q squared is 450 out of 1,100. So I always have to make sure I always start with Q squared. I'm going to do the same process again. So I'm going to square root Q squared to find Q, which is 0.64. So now I can use that formula P plus Q equals 1, rearrange it for P, so that's 1 minus Q, and then I'm going to be going 1 minus 0.64, and I'm left with 0.36. So that is my allele frequencies, but it's asking just for the genotype frequencies in this question. So I have to continue going with that really big formula, put in my information. So I know that P is 0.36, so I'm going to square that to find my homozygous dominant individuals. My heterozygous individuals, I'm going to go 2 times 0.36 times 0.64. And then I end up with this information. So again, you're going to make sure that these three numbers add up to 1. Otherwise, you've done it incorrectly, and I made sure. So that means that 13% of your individuals are homozygous dominant. That means that 0.46, so that's 46%, are heterozygous, and 41% are homozygous recessive. So those are your genotype frequencies, and that's how we solve the second question. So why don't you guys try the other questions on your own, and I'll give you the answers in a little bit. Hopefully it makes sense, but if not, I will answer all your questions in class.